Alright guys, welcome back to Underground Science, and in this video we're going to be looking at the oxidative phase of the pentose phosphate pathway. Alright, so one thing to know about the pentose phosphate pathway, right, so the phosphate pentose phosphate pathway actually occurs in our cytoplasm of our cells, right? So we're going to look and mostly in our liver cells actually also. So if we look at how the pentose phosphate starts, if we take our liver cell, for example, remember glycolysis, guys? So remember glycolysis, we went from glucose. So this is our liver cell, and that we're going to be talking about the cytoplasm of our liver cell, all right? So cytoplasm of our liver cell. Great, so remember glycolysis. So let's remember back to glycolysis. What happened in glycolysis? So in glycolysis, we actually broke down glucose into three carbon structures, right? Two pyruvates. So if we recall, we went from glucose to glucose 6-phosphate, right? We went from glucose to glucose 6-phosphate, and we actually used the enzyme. If we're in the liver, we use the enzyme glucokinase, right? Glucokinase, all right, to catalyze this reaction between glucose to glucose 6-phosphate. And we also remember that we have this phosphate here because we break down ATP. We use an ATP molecule to break it down into ADP. Oops, sorry about that. We break it down into ADP and an inorganic phosphate, and then this phosphate actually attaches itself onto the 6-carbon sugar, onto the 6-carbon of glucose, making glucose 6-phosphate, right? But from here, what can happen is that, so we already reviewed glycolysis, right? But from here, what can happen is that instead of going from glucose 6-phosphate to fructose 6-phosphate, then fructose 1,6-bisphosphate, and all of that in glycolysis, the pentose phosphate pathway actually diverges from here. So from glucose 6-phosphate, we can actually start a pathway, especially in the liver, called the pentose phosphate pathway, which is what we're going to talk about in this video, but in particular, we'll talk about the oxidative phase of the pentose phosphate pathway. I don't know what I was writing there, sorry about that. Pentose phosphate pathway, all right? So the starting molecule of the pentose phosphate pathway is actually glucose 6-phosphate. So we can pretty much take another turn, right? We're driving on a street, right? You could think of it as we're driving on a street of our glycolysis street, but once we get to glucose 6-phosphate, we turn on another street, and we can do the pentose phosphate pathway. Now, there's actually, I said two, but let's just look at some of the main purposes, right, of the pentose phosphate pathways to make this molecule called NADPH. Okay, so especially in the oxidative phase, we make a molecule called NADPH, and this is really good for especially the liver because our liver contains high amounts of glutathione, which is a very important antioxidant that our liver uses to detox certain compounds, right? To detox certain dangerous things. And so glutathione needs NADPH, right? We discussed this in the glucose 6-phosphate dehydrogenase anemia, right? Our liver, or right, especially particular liver as an example, needs NADPH in order to, right, get the glutathione back to its reduced form. Okay, but if we don't have enough NADPH due to a deficiency in our glucose 6-phosphate dehydrogenase enzyme, we saw that we can get G6PD-H anemia. Right, so if you haven't already, go check that video out, all right? So one main purpose is to make NADPH, okay? And the other main purpose of the pentose phosphate pathway, and there can be more, but we'll just focus on these two, right? Especially this NADPH formation in the oxidative phase, all right? But anyway, the other one is to form a 5-carbon molecule called ribose 5-phosphate. Ribose 5-phosphate is really good. We need it to make our nucleotides, to synthesize our nucleotides in our RNA and DNA, all right? Because remember, a nucleotide is made up of a phosphate group, a 5-carbon sugar, right, or a ribose sugar group, and a nitrogenous base. Okay, so we need ribose 5-phosphate to make nucleotides that are present in our RNA and DNA. Okay, so let's go and look at the oxidative phase of our pentose phosphate pathway. So let's go ahead to another page and start off to glucose 6-phosphate. All right, and if we want, we can talk about this as being it's occurring in the cytoplasm of our liver cell. All right, so let's go and write that neater. Sorry about that. All right, so let's go and take a look at this. So we start from glucose 6-phosphate and we convert to another molecule called 6-phosphoglucano. Hopefully I'm spelling all this right. 6-phosphoglucanolactone. Okay, 6-phosphoglucanolactone. And we use an enzyme here. Okay, so the enzyme that's used here in the conversion between glucose 6-phosphate and 6-phosphoglucanolactone is going to be glucose 6-phosphate dehydrogenase. All right, so glucose 6-phosphate dehydrogenase. Again, hopefully I'm spelling all this right. Okay, so remember that this enzyme is super important because it helps make NADPH. So again, what happens in this step, from the conversion of glucose 6-phosphate to 6-phosphoglucanolactone, our glucose 6-phosphate dehydrogenase enzyme actually drives the reaction of making a NADPH molecule. So what happens is that we have an NADP positive turn into NADPH. All right, so we have our NADP positive molecule gets reduced and we have an NADPH molecule. So we form a NADPH molecule, and if we see a deficiency in this enzyme, it can lead to a decreased amount of NADPH formation, which can then have we can see some trouble that occurs between our glutathione, right? We need reduced form of glutathione, and we'll see that it's actually the NADPH that helps reduce glutathione, and glutathione is an important antioxidant. And if we have a deficiency in NADPH, glutathione can't be reduced and can't do its important functions, which means that if you saw in the glucose 6-phosphate dehydrogenase anemia video, it leads to the destruction of the hemoglobin, right, in our red blood cells. But anyway, this is what happens in the first step of our oxidative phase of our pentose phosphate pathway. Next, what happens is that our 6-phosphoglucanolactone gets converted into 6-phosphoglucanate. All right, 6-phosphoglucanate. All right, and so in this step, what enzyme is used? We use a glucanolactonase enzyme. Okay, so let's go and write this down. So in this step, we use a enzyme called glucanolactonase. So glucanolactonase will be the enzyme responsible to catalyze the reaction of 6-phosphoglucanolactone converted to, so 6-phosphoglucanolactone will be converted to 6-phosphoglucanate via glucanolactonase. All right, so actually I should write this in blue so it's not that confusing, okay? And then in this step, we actually use a water molecule. So we use a water molecule, but then what comes out is a hydrogen cation. Okay, so I'm going to draw a better looking arrow. Sorry about that. What comes out is a hydrogen cation. And that's pretty much what happens in this step. Next, what we see is that 6-phosphoglucanate gets converted into ribulose 5-phosphate. All right, so ribulose 5-phosphate is now a 5-carbon molecule. We went from a 6-carbon molecule to a 5-carbon molecule. That means we had to lose a CO2, right? We had to lose a carbon, but in the form of carbon dioxide. So let's go and write that out. So we actually lost a carbon dioxide in this step, a carbon dioxide molecule, because we went from a 6-carbon molecule to a 5-carbon molecule. Okay, and in this step, the enzyme used is 6-phosphogluconate dehydrogenase. All right, so the enzyme used, let me go and write it down, I guess, in orange. So the enzyme used to catalyze this step, or you know what, because it's already full, let's just go ahead and write on the other side. So the enzyme used to catalyze this step from 6-phosphogluconate to ribulose 5-phosphate, we use 6-phosphogluconate, right, dehydrogenase. Dehydrogenase. All right. Okay. And again, because we see a dehydrogenase here, that should indicate the fact that we actually make another NADPH molecule here. All right, so we actually have an NADP positive that gets reduced and turns into NADPH. All right, so remember, this was one of our main purposes to the pentose phosphate pathway, right, of the pentose phosphate pathway. So we made NADPH here too. So let's go and erase this arrow because we're not talking about anemia right now. Okay. All right. So, and this right here, after this, we're going to see that ribulose 5-phosphate will actually split into two molecules. And from here, we're going to have the non-oxidative phase start. All right. So the non-oxidative phase we'll do in another video. All right. And we'll see how we use an epimerase enzyme and a isomerase enzyme, and how the non-oxidative phase continues. All right, and actually in the non-oxidative phase, we make a lot of compounds that actually can be used in the glycolysis pathway. All right, and we'll see all that later. So the main purpose of the oxidative phase, or the aerobic part of our pentose phosphate pathway, so let's go and write this down. This was the oxidative phase of our pentose phosphate pathway that was occurring in the cytoplasm. We took the liver cell for an example. We made 2 NADPH molecules, all right? And this will be really important when we talk about, or actually we talked about it in the anemia video for G6PD-H anemia, where NADPH is really important to help glutathione function. All right, glutathione is a really important antioxidant. All right, so this is it, right? This is it for the oxidative phase of the pentose phosphate pathway. I hope this video made sense to you guys. I hope to see you guys in my future videos. We'll talk about more cool topics about biochem, physics, and chem. All right, and maybe other subjects too. Don't forget to hit that notification bell and like and subscribe, and see you guys later, and stay safe.